by instructions from Karen. I am the president of the Houston chapter, have been for quite a long time, and I'm very proud of that. And have been involved with the Technion. I'm on the ATS National Board and on the International Board of Governors, and have been for a while. And probably the most, the highest distinction I have altogether with regard to the Technion is that I actually taught a course there 3 times, a short course. By very happy coincidence, I happen to have some expertise in deep water drilling and production. And the coincidence was that Noble Energy found some natural gas, as you are all aware, I'm sure, in the Mediterranean off the coast. And there weren't any petroleum engineers or deep water engineers in Israel. So the Technion decided they'd better institute a course and teach about that so that they could develop some engineers. And I got a chance to go there a few times and share my expertise with some of the students on the campus. So that's what I was talking about on those picnic tables there that you see behind me, where I would eat my lunch for that 1 week. They made it a 1 week course. And with us today, we have 2 of the staff members of the Technion, regional directors, I should say. And that's probably not exactly the right title for each of them, but it'll do. One of them is for Houston, and that's Karen Weiner, excuse me. And the other one is Robert Rothschild, who's in Los Angeles, but also covers Dallas. And I've known both of you for some time. So thank you very much for all you do in supporting the Technion. So my job now is to introduce our illustrious speaker, and he truly is illustrious. I've known Assaf Zinger for some time, ever since he came to Houston. And I will say it was fortuitous that he came to Houston, and we're all very happy. He's been here 2, almost 3 years, something like that. Yeah, so terrific. He has 3 degrees from the Technion, so that shares with the illustrious background that he has of being in Houston and also working in research, postdoctoral research at the Houston Methodist Research Institute, where he's developing personalized, now here we go, biomimetic nanotechnology, which means inspired by nature, platforms for drug delivery and regenerative medicine. I said he has 3 degrees from the Technion, a bachelor's degree in biomedical engineering, a master's and doctoral degree from the Faculty of Chemical Engineering, where he did his research work under Professor Avi Schroeder, who's been here in Houston a couple of times, wonderful guy and very, very accomplished. And that work has been on targeted drug delivery and personalized medicine technologies. He has also been very active in his undergraduate years at the Technion, and maybe he'll tell us a little bit about that later on. He's the grandson of a Holocaust survivor. And he's been, of course, like all healthy Israeli men and women, for that matter, most of women, but all men, served in the military. He served in the armored brigades and has fought in 3 wars and is still involved, as most young Israelis are, for how long? 15 years of reserve duty or something like that. After now it's 2.5, but probably when he was in it, it was 3 years of active duty. So it's a total of 18 or more years that these people serve. It's utterly amazing what they give to the state of Israel for the defense of the country. He is, and he's also still active as an executive officer in the Armored Corps headquarters. He lives in Houston, we're happy to say, for over 2 years with his wife, Noah, who is an occupational therapist working with autistic and deaf children, and is manager of the Gvanim program at the Israeli American Council. And they have 2 daughters, Ayala and Tamara. And we are so delighted to have you with us. Thank you very much for being here, Assaf.
Hey guys, sorry, howdy guys, howdy all. Thanks for the very kind and nice introduction, Terry. We known each other since I was, I think, I would say 25, right after the army when I met you and Larry Jack here in Haifa, after Protective Edge. And look what happened. Look what happened. No, I'm kidding. That's a great honor. And actually, I pretty like this setting. So we have 10 people. So basically, let me entertain you guys. So please don't be quiet. Stop me in the middle, ask questions. I prepared 2 stories for you guys, and I don't know how much time I have, but Karen, you say 30 minutes, right? 35? Good. But please, if it's boring, tell me yourself this story is boring, and I will jump to the other one. Basically, no. And Terry, you know me. I'm in Arabic. We say, "Let's talk Dugri." Dugri.
You have a talent for making what may be otherwise boring into exciting. So I have no trepidation about this.
So in Arabic we say, "Let's talk Dugri," which is like, let's be honest, let's be blunt. And this is why I really like the Lone Star State, because they call me the Israeli cowboy. I basically say whatever I want to whomever I want. When we did the orientation in the hospital, small joke, and then we started talking about science, you're going to Methodist hospital orientation. So one of the scenarios in active shooting scenario in a health caregiving facility. So what they teach everybody is to run and hide. So I raised my hand and I told them, wait, I'm taking over and I'm running to attack. So I literally had a talk with my manager that I'm not allowed to do it, by the way, just for all of us to know. So since then, I'm behaving. Okay, so as Terry kindly I'm introducing, my name is Assaf, PhD from the Technion, interdisciplinary research, biomed and chemical engineering. I came to Houston because my wife told me she didn't want to go to Harvard, MIT because of the weather. And I told her, you know what, you're right, and we love it here. We actually love it here. I fell in love with this state. Tamara, as Terry said, was born here. So give her 25, 28 years, she will be our president, just wait. She can be our president. She was born on Texan soil. We are Rockets and Texans fans, and honestly, I still did not understand how baseball is being played, but let's speak after that. Okay, so I'm sharing my screen. Let me know if you can see it. Very nice, enhancing inflammation, so on and so forth. And as I told you, please stop me. So I can see your faces on the side. Please feel free to engage or send text messages. So the first story will start with just a little background. Why did we choose the medical center? So Houston has the largest medical center in the world, which I did not know before I came here. If you will read this data, you will basically understand that if someone wants to do translational medicine, he needs to come to Houston, Texas. And that's exactly what I missed in my pillars when I graduated from the Technion. So I had my very strong engineering basics, but then, you know, we developed these amazing tools, but we never managed to inject them or test them on human beings. And this environment was given by Houston. And basically that's what I learned here. And, you know, one day I'll bring the Torah back to Zion and I will teach them in Israel how to do it right. Because everything is bigger and better here, right? Why do I want to go back? Because that's Israel. So, you know, we are very small. Apparently, I can drive in Israel 5 hours from north to south, but in Texas, I need to drive, I think, 12 hours in order to just get out from Texas. But again, Israel is like entrepreneur leadership all over the world. You can just read the numbers. So I think these two combination of the Lone Star states together with Israel is very meaningful and impactful combination. So the first scientific story of today will be the story of pancreatic cancer. And I don't know if you knew that, but pancreatic cancer is the most lethal of all cancer. Key statistics, it's awful. It's like 7% 5-year survival rate, and it's the 4th leading cause of cancer-related death in the United States. And I don't know if you ever saw how pancreatic cancer look like. So let me be the first one to introduce you. So this is a healthy pancreas, and this is a diseased pancreas. And even if you are not certified as a physician, you can see these big islands that are basically not related to the tissue. The specific staining I used called Masson trichrome staining. This staining basically stains collagen fibers by the color of blue. So every time you see blue color, you can say, hey, Assaf, this is collagen. So it doesn't say too much, but if you just dive in deeper, you will be, now you can actually notice the differences. And if you look at the healthy tissue, pancreas, compared to the diseased one, you can see that the blue fibers, the collagen fibers, are all over the place. So just touch your ankle for a second, your Achilles tendon, and this is basically what we are being reported in the surgery room. If you touch this kind of tumors, you will actually feel a rigid tissue, like a tendon, like tendons in your leg. This thing by itself caused very strong pressure on the blood vessels and basically act as a bunker that prevent chemotherapy or any other drugs to penetrate the tumor itself. So during my PhD, I actually developed a program that can assess the difference in the collagen burden between diseased and healthy pancreas. And we found out that the differences are almost 12.25-fold denser between the two. So as Terry mentioned, I'm a major in the Israeli Defense Force. I was the company commander of a tanks during Second Lebanon War and after it. And I decided that I need something like this. I need something that can actually breach the bunker. And if you see the plane as a tumor, you can see that the chemotherapy penetrates and basically demolish the tumor. But things happen. And for a long time, we failed. So for a long time, I've been injecting this, paclitaxel, gemcitabine, tons of awful toxic drugs to these poor mice without any good results. And then protective aid started. So basically I stopped everything and went down to Gaza because I'm the first one who recruited when my company has been recruited because I'm the executive officer. And during the fight in Gaza, in this very stormy night, I found myself on this sand dune, sitting with my soldiers. And then it's the first time I understood that Hamas can dig tunnels. And it hit me. And don't tell it to Hamas, but basically they gave me the inspiration to produce, as I call them, Hamas nanoparticles, but as we really call them, we call them enzymatic nanosurgery. So basically what I decided to do is to take collagenase, an enzyme, and actually encapsulate it into a nanoparticle. This enzyme, collagenase, know how to disassemble collagen fibers, the same bunkers that actually surrounding the tumor. And this is what I did. So basically I wrote a patent on how to encapsulate collagenase, nature's scalpel, right, into a nanoparticle. And as you can see in front of you guys, I managed even to tweak the way we will release this enzyme. Because I looked at our body and I understood that if the membranes of the cells in our body has a higher content of cholesterol, there will be a bit rigid. So adding cholesterol to the Hamas nanoparticles, I basically can control the release of this collagenase. Why it's important? Because when the collagenase will arrive to the bunker, the pancreatic bunker, it will make small holes that will allow chemotherapy to penetrate. So that's what the idea, inspired by Gaza, as I told you guys. And as you can see on tile A, you can see like a pancreatic tumor. The white part in the tumor where my laser, can you see my laser?
Yes.
Perfecto. So when you can see this laser, this is the tendons we just mentioned. Then I injected fluorescent nanoparticles, and then you can see they did reach the tumor, but in very low concentration. Less than 1% of the particle reached the tumor. Why? Because of the bunker I just told you, right?
How did you know that it would penetrate?
That's a good question, but this is all science, right? I had no idea. I had a solid hypothesis. And as a good soldier, I described the battle. So I know I have a lot of collagen. I knew I have blood vessels. As you can see, I have blood vessels in the tumor. Yes. So basically, if I inject systemically particles, they should reach through this small blood vessel to the tumor. The low percentage accumulation actually reflect the fact that something blocked my particle from being accumulating in the tumor. So I expected 90% accumulation and I saw less than 1%. So what I did next, I basically collected the biopsy and my particles are in red now. And my biggest fear was approved. I basically saw that my particles, the red dots, are only in the healthy pancreas. They never breach the tumor. So now I knew I have something in my hand. So my particles reach the pancreas, stuck in the healthy tissue. And now basically we need to let them walk, right? So these particles need to walk a bit. So what I did, I collected 3 biopsies and I soak them in the collagen as nanoparticles. I compare the collagen burden of the tumor itself, not treated, with a healthy pancreas, and tumor treated with my particles, and I managed to prove, that I managed to decrease the level of fibrosis, the level of collagen fibers, the same blue I showed you in the first image, almost to the level of healthy pancreas. And remember, this was ex vivo, out of the animal body, made just to test a second before we inject it. The next step was actually to look what happens to the tissue. So we decellularized the cells, we actually removed all the cells from the tissue. And as you can see now, we basically got the same holes, as I call them, the same burden relief as the healthy pancreas. And you can compare it to the same pancreatic tumor dense matrix. So the next thing was to do an in vivo model. So we took pancreatic tumors that can express red fluorophore. This is the red dot you can see. And basically, if we follow this red growth, we know that the tumor is propagating. If this red fluorescence will decrease, we know that our treatment caused something to these animals. So what we decided to do was to inject our collagenase encapsulated nanoparticles twice in order to relieve the burden of the fibrosis. And then we injected paclitaxel encapsulated in mice cell. So paclitaxel is a chemotherapy. And we monitored the animals for 14 days. And after 14 days, we were really happy, basically, but super excited to see that in the control group, treated only with the paclitaxel, the tumor basically grew, while in our control groups, our test groups, when we injected our particles followed by the paclitaxel, the tumor almost disappeared. And we were super happy about that. So basically, we reached the pancreas, we made some holes, and chemotherapy can penetrate now and treat the tumor itself. However, one of the nice reviewers in this journal wrote me, Assaf, listen, you basically touch a tumor that nobody wants to touch it. Because once you poke a hole in the tumor, you allow tumor cells to circulate in the blood and actually cause metastasis. And at the beginning, I told you, you know what? He's right. So basically, we cure the tumor, but kill the patient. But then we started to check what is the size of my particles. And the size of my nanoparticles are 100 nanometers. And if you will know that tumor cells are 20 micron, you basically can hypothesize that these cells cannot go out from the small holes we are making. And that's exactly what happened. So basically, we compared treated mice with non-treated mice, and we didn't find any evidence of metastatic cells, neither in the blood or in the filtering organs. Of course, we did some pathology staining to prove that all the filtering organs are intact and the same, and we did not see any differences. So basically, this paper was published and offered for the first time a way to treat pancreatic tumors without a surgery. And you know, when I'm bringing this hope and happiness to the world and telling, yeah, we can cure cancer, it's really important to say, guys, we did it on mice. The time that take for this kind of therapeutics to translate from the bench to the bedside can take up to 10 or 12 years. We still don't know, I'm talking about humanity, how to make this process faster. And this is why I came to Houston. And I will tell you all about that. Robert.
Question. Fluorouracil was one of the chemos that they administered back in the 70s and probably for many years before and many years after. But that was never a direct injection. It was actually, they didn't have the smart drug delivery. Would that have had a chance, that kind of a drug back in those days, had we had the means that you have today? Or was that just a bad drug to begin with?
I'm going to tell you the truth, right? We're not recording this talk. Thanks to the FDA, we have a lot of barriers now. And I'm saying thanks, and I'm being cynical from one hand, but on the other hand, I know it comes to protect humanity, so I appreciate it. We still use drugs developed in the Second World War II. Because the time it takes to translate drug from, as I said, bedside to clinic can take years. Just for example, a few weeks ago, I started to remanufacture punatinib. Punatinib is also fast approval drug that was approved for osteosarcoma patients. The only problem with this drug, Robert, when you inject them systemically, you kill all the cells. They don't know how to distinguish between healthy to diseased tissue.
Mm-hmm.
And I don't know if you heard the Nobel laureate, Professor Duadana, the professor who discovered the CRISPR-Cas9. She said, guys, we created worlds, but we don't know how to deliver it. So it all comes to delivery. And basically every drug that you can encapsulate in a particle, you can deliver. The challenge is to where. And in this case, we tried to use paclitaxel because gemcitabine, for example, was not effective. Okay, any other question before I jump into the second story?
Yeah, do you have to go directly from mice to humans, or can you do tests on large mammals that might that might help you on the way, might speed it up?
No, you're right. So basically, FDA demands to do large animal models before going to phase 1. So basically, the data I just showed you, called in the perspective of startup, venture capital, preclinical data, the next step will be safety, which we can start to do it in large animals, and then we move into phase 1, which are humans. This stage, the mice versus large animal stage, in 2020 is still a huge roadblock. If you will count the number of papers, scientific papers that were published using large animal models, you will find very few, unfortunately. The reason, and I'm telling you that because I'm now conducting large animal models trials, is first of all, the money. Large animals are cost almost tenfold more than small animals, than murines. And then the handling itself. So I'm working with specific kind of animals that just to touch them, I need to go to a training of 6 months because, you know, they are federal protected.
Yeah.
Which I also respect. But this is very important. If we will find a way to bridge this gap, how to take our formulation and push them forward, things will go much faster. So there is a new initiative now that basically try to replace animals with chips. It's called lab-on-a-chip. So basically they try to mimic the way our biological system are working. And instead of using animal, you can use a chip that it will yield the same result. However, it's very it's in its diaper yet. It's a very nice new initiative, but it will take some time. So you know what I did? I basically told the Technion that if they want me back, I'm going to get co-affiliation with Methodist. So they know how to translate research to humans. So every time you write patent at the Technion, you just deliver the formulation and they inject it. But let's see.
Assaf, I'm curious if you can speak to whether it's any faster in Israel to do this kind of research than it is in the United States. I know, you know, the FDA tends to be, you know, more conservative maybe than some other places. Can you fast track it more in Israel or no?
So I would say like that. So the European developed these shortcuts that called the 510K. So instead of going to a full FDA, tons of money, 5 years at least, so on and so forth, you can basically say that somebody developed something similar and you're riding on this wave. Still, it will take 3 to 4 years in Europe. And I think what people start to understand now, and I'm not among these people, I'm very young, so be very cautious on what I'm saying. Asia. So basically, we start to understand that Asia jumped this barrier very fast. And a lot of people are starting to invest in venture capitals in Asia during the understanding that they cannot use their drugs in Europe and in the States. So it depends, right? I think it's ethics code, if I may say. So do you want to do it without the needed regulation? So I will say no. But again, maybe I'm young and I still need to learn something.
Yeah.
But, and you know, I will also add, so Karen, just imagine, that in order to reach to MD Anderson or to Methodist Hospital, I basically need to walk for 2 minutes. So I basically can hold my nanoparticles in my hand and walk 2 minutes and I'm in the clinician room, right? In Israel, in order to cross from the Technion up in the mountain to Rambam or to the Carmel Hospital, we are talking about almost 40 minutes drive in traffic. And that's time. So I told my wife, if we are going back to the Technion as a PI, I'm going to buy a motorcycle because I want in 10 minutes to get back to the hospital. I cannot, and this is something I have to teach my students, I cannot leave the connection to the patients. When I'm walking to the medical center at morning and taking the number 4 bus, I'm sitting next to cancer patients. I'm eating in the cafeteria with cancer patients, cancer survivors. So I'm inspired by these people, by their strength. And basically, I told no, I think I'm working better when I understand the need. So I don't know how to bring this need to the faculty, but I have several ideas. So for example, every conference that talk about cancer nanotechnology should start with a cancer warrior that stand on the stage if he wants to share his story, just to inspire us, because I really believe in motivation driven by inspiration. But thank you, super good question. Yeah,
Thank you.
I became motivated just for talking about that. But yes, that's the truth.
Good.
Okay.
Okay, go on.
Next story. And this is more COVID-19 related. I removed some slides, and if we would have time, I will definitely show it to you guys. As long as we're not being recorded. So now coming the cool stuff, right? So I learned how to make these nanoparticles, so on and so forth. And then I started to ask very annoying questions. So I don't want to treat animals like a black box. I don't want to make the nanoparticle on one hand, inject them and see what happens, right? It's very boring. So I decided that I want to, close my eyes for a second and imagine I am the nanoparticle. And again, let's put the scale. So nanoparticle is like 10 in the order of -9 meters. It's like super small. Cells in your body are 10 to 20 micron. Hair on your head is 200 micron. So just for you to understand, we're talking about 6 order of magnitude difference between these small red blood cells to my nanoparticles. So what I decided to ask myself is, can I learn from the cells in our body how to target inflamed tissue? And then basically hijack it to my nanoparticles. So no more, Terry, synthetic nanoparticles that you inject in vivo and go to the liver, but nanoparticles that basically act like white blood cells. In order to answer this question, again, like a good soldier, I dissect the inflammation mechanism. So when tissue injury caused in your body, white blood cells are recruited. But why? Why? How do they know to reach the inflamed tissue? In order to answer it, you have to look on the cells themselves. So if you will ever talk to a white blood cell, the first thing you will see on him will be LFA1, which is a lymphocyte function-associated antigen. This is basically a membrane protein that is present on the white blood cell's membrane. And all he's doing is detecting inflamed membrane proteins on endothelial cells. Why it's important? Endothelial cells are the cells that actually build your blood vessels. So basically, when a blood vessel gets inflammation, there is this beaker of a protein, ICAM1, that is raised, and then white blood cells that normally, statistically, just circulating in your blood, adhere to it. This LFA1 to ICAM1 connection is something that I try to hijack. So I told myself, again, a scientific hypothesis, if I manage to steal from white blood cells their LFA1 receptor, I can target inflammation. And why it's important? Because I can put anti-inflammatory drugs like dexamethasone that everybody talks about in the COVID-19 now, and basically not only target inflammation in the lungs, also deliver anti-inflammatory drug. So that's what my hypothesis. And to make long story short, I already published this. I basically took white blood cells, steal their membrane protein, and assemble a nanoparticle. So the same nanoparticle I did in my PhD, but this time I gave him some, if I may say, badass missiles to fight inflammation and target it. And this is how you can see in front of you guys. So this procedure takes 2 hours since I'm collecting the patient blood. So basically I'm collecting from a patient blood, I extract the white blood cell, extract their membrane protein, and basically encapsulate anti-inflammatory drug. And now these weaponized nanoparticles can target inflamed tissue and release anti-inflammatory drugs. And we got a nice cover in Circulation Research cover because now I call them my Lego nanoparticles. Because Karen, you can come and tell me, Assaf, listen, I want this membrane protein on the particle with this drug. So I basically assemble it for you as a good engineer that understand what happened in our body. And of course I fine-tuned it. So how can I improve the inflammation targeting? we just add more LFA1, the same beakers that target inflammation. So this is what I did. I basically developed a method to extract membrane protein from white blood cells. Sorry, as you can see in front of you. And what I did, I tested it in two different models. I tested it in gram-negative bacteria inflammation, local inflammation, and in triple-negative breast cancer. So this is actually a cryotransmitting electron microscopy of my nanoparticles. And as you can see, when you add more membrane proteins, you don't see any difference between the particles. The only thing that change a bit is their charge. This is an evidence that I added more membrane protein. Then I did some Western blots, which are actually antibody way to detect proteins on my particles. And as you can see, the CD11B and CD11A are the ones who build the same LFA1 I told you. So as you can see, I basically increased the concentration of this protein on my particles. Then I took endothelial cells in vitro in my lab, and I inflamed them using lipopolysaccharide, the same sugar that inflame tissues in our body. And as you can see, the inflamed endothelial cells uptook more of my nanoparticle. This is the red color. So basically what I just presented in front of you, and this is an IP of Texas, what I wrote for Methodist, I developed a nanoparticles that can target inflammation like white blood cells are doing. After the in vitro, I started to use intervital microscopy. And I will quickly explain what is intervital microscopy. So basically what you see in front of you is live animal. So this animal is alive, just anesthetized. And what I did, the microscope basically scanned blood vessel inside the animal. So Gary, you asked me, Terry, I'm sorry, you asked me at my fourth slide, how do I know the particle reached the tumor, right? So look what I did. I basically imaged my nanoparticle for you. On green, you can see blood vessels. In red, you can see my nanoparticles. And look what happened. You can basically see my nanoparticles circulating in the live animal blood. This is what you see in front of you. And the interesting thing is that my leucosome, leukocyte biomimetic nanoparticles circulate much better than ordinary synthetic nanoparticles like I did in my PhD. So basically, weaponize the nanoparticles with white blood cells, membrane proteins, give them not only the option to target inflammation, but also to evade secretion from the blood. So we were super happy about this result as well. And Terry, now you really believe me when I say my particles are in the bloodstream. We tested it in inflammation, local inflammation induced in the ear of mice. And you can see that the leuco 1 to 20. So the particle with the highest concentration of proteins basically reach the inflamed tissue better than the synthetic particles, which we call liposomes. And not only in this model, but also in the cancer model. So you can see how the white blood cells like nanoparticles target tumors better than the normal synthetic nanoparticles. And of course, we checked toxicity for these particles. We did not notice any toxicity of these nanoparticles because basically we said, you know, there are like white blood cells. So what about FDA? So this paper is now under revision in ACS Nano, which is a very decent journal to publish with. I think it's ranked second in our field after Nature Nanotechnology. And again, you can ask me, what about FDA approval? So I can tell you that Methodist took them almost 2.5 years to write this patent because they actually protected everything. And I can share with you now that I'm basically doing large animal models with this kind of nanoparticles. And if this is going to work, I think the transfer technology office will probably going to sell it to some venture capital. And you know, if I will be in Texas, I don't pay taxes here too much, so it's good. But if I'll be in Israel, it's a whole different story. Any question about this story? Oh no, was it clear? I don't know. Did I speak too fast?
No. This is just wonderful information, and you covered a lot of very scientific and interesting subjects. I'm wondering if all of you who are with us, if anybody has any questions they want to ask. This is our opportunity to have conversation with Assaf.
I guess I'm wondering, sitting here wondering what kinds of cancers obviously not blood type like leukemia, but what kinds of tissue cancers can be treated by this method?
So Terry, you know what? I'm going to tell you the honor. I'm going to be very blunt as always. When you ask a researcher why he chose a cancer model, there are basically two answers that I heard in my life. The first one, someone in the family was sick, and I just decided to fight this tumor. The second option is federal funding. So basically the DOD or the NIH publish, in Europe it's the ERC, publish a call that say we call researchers to investigate this cancer because of, I don't know why.
Right.
And this is why the research is derived. So what you are basically, what I'm basically telling you is I chose these models because they fit my hypothesis. So triple negative breast cancer is a well-known cancer that inflamed blood vessels. If you will ask me, Assaf, can it also target osteosarcoma? I will tell you maybe, I don't know, right?
Okay.
So, you know, and I'm not trying to affect, but I think that, again, a lot of time people ask you, why do you choose this model? So first of all, it's like personal story.
That isn't what I asked you because I didn't know enough to ask a question like that. But what you're basically telling me is you choose to address a certain type of pathology because that's what there's money available to study. And in order to answer my question, you would have to study other ones and find out whether or not it works. It isn't obvious. Is that right?
But I mentioned something before that. So I told maybe personal motivation. So somebody related to you, and then I will say the funding. And I think in this case, there is a lot of power to the funding body. So I don't know who decide what to do or which scholarship or fellowship to call it, but basically that's a very strong position to decide in which direction a research will go.
Yeah. Okay.
And by the way, Mary, you want to ask a question? You have to unmute yourself. I cannot do it.
Marilyn, you go ahead.
Can you unmute her?
Okay.
There you go. No.
I'm sorry, Mary, we cannot hear you. You have to unmute yourself.
Marilyn, unmute yourself.
Trying, but there you are.
Oh, okay.
Hear me
Okay. Now we hear.
I can't remember what I was going to ask. Oh, yes. So which cancers are you directing this last presentation?
So the first one is triple negative breast cancer. The second one is osteosarcoma, because I got request for a family to pursue this kind of cancer, and unfortunately, it only affects pediatric population. So I decided to do it as another effort. But what I'm going to say is maybe very radical and very non-responsible. Every cancer that in the biopsy, we can see inflammation in the blood vessels, these particles can treat. So I will leave it that big. And I actually have several other slides if we have time or not, just stop me, to show you how basically I'm going to tailor it to COVID-19. So I never thought I will do COVID-19, but basically we talk about inflammation in the lungs, right? So blood vessels are inflamed. So basically you can take these nanoparticles, put the same anti-inflammatory drug, and basically treat the lungs. And if I will establish my new lab in several months, that's probably one of the biggest projects I will go, because everybody will pay money for me to do this kind of research, right? And I will also add that, you know what, just because we have time, like I'm sharing let me steal you guys. If you already like spend some time with me, so let's do it interesting. Let me show you something really cool. Wait. Okay. You cannot see my screen yet, right?
No.
Okay. And now you can see my screen.
Yes.
So basically what I'm going to show you, it's a patent written way to make genetic cargo encapsulated nanoparticles, okay? And I will say in a second why it's important. So basically what you see in front of you is a very robust system to encapsulate genetic cargo, which can be mRNA, siRNA, so on and so forth. Spoiler, these things are being treated as vaccines today inside the nanoparticle. So in the green you can see the genetic cargo. And then you can see actually my nanoparticle assembled around it. Why it's super impressive? Because what I show you in one nanoparticle can be translated to almost 10 liters of nanoparticles, right? And if we think about vaccines in the future, so basically what we want to do as a study effort, and I'm going to replace, again, do it very fast because we have time. We want actually to develop vaccines. So we will need particles because I told you, delivery. We want to encapsulate genetic cargo. And in this point, I will say we need help of biologists, which we have. And what we want to do now, and again, this is super new data, so don't judge me. Wait. You can see the red things on my screen, right?
Mm-hmm.
What you see in front of you are cells. In red, you can see the nanoparticles, and these nanoparticles encapsulate genetic cargo. If the genetic cargo will be released in the cells, these cells will start to express green fluorescent protein. This is the marker for me, that I cause the cells to produce the protein. And as you can see, after several hours, you can see these cells starting these green beakers. So if you think it about COVID-19 way, so I will take genetic cargo that can basically express the corona of the COVID-19, the same crown of proteins he has, and basically cause the body to manufacture antibodies against it. And this is preliminary data, still writing the patent, guys. So if you tell somebody I messed the novelty, I will deny. That was a friendly talk, so I'm not worried about that. But again, I don't want to be too optimistic because I don't want to lie to you. The main roadblock that Moderna, Pfizer, and even the smaller stuff in Texas are handling, when we inject this particle, they go everywhere. So not only specific cells express this corona, all the cells in our body. And now comes the question about immunodiseases. Are we going to cause the body to attack himself? And this is why we don't have vaccine. Hopefully, somebody smarter than me or one of my students will manage to develop a very useful targeted tool for this mRNA. And I think you already got that. We know to target inflammation. All we need to do now is just to put the right genetic cargo inside it, and then only the lungs endothelial will express this peptide. So give me like 7 months and I'll tell you if I succeed.
I remember that they said that one of the presentations, Marcel Makhlouf was working on something similar to this. Is it similar, or the kind of delivery system, nanoparticle delivery system, is that similar to the research you're doing?
You know what, I'm going to ask you an opposite question. If I will tell you that I know 300 people who dig oil in the sea, are they doing the same thing?
A lot of people are working, okay. But I happen to know her, and that's why I wondered.
No, I love Marcel. She's one of my mentors. Her NanoGhost in the 90s were a hit.
NanoGhost, that's right.
NanoGhost. She basically established company. One of the differences is that she's using mesenchymal stem cells and not leukocytes.
Okay.
So that's a different niche. And she's using biological lipid. So basically she's taking the cells as themselves and encapsulate stuff in them, while I'm using lipids, which as an engineer, I choose and put whatever I want inside. So pros and cons for two approaches. But Terry, if you compare me to Marcel Makhlouf in the age of 36, I'm going to retire.
I didn't mean to make a comparison, and it was just a very, very ignorant type of question.
That's a compliment. That's a huge compliment.
Okay.
Thank
All
you.
Right. I have another kind of simple-minded question. I know that corona viruses are tiny, and it's only the last 30 or 40 years, I guess, that we've actually even been able to see them because of the magnification issue. How does the size of a coronavirus compare with these nanoparticles?
That's also a very good question. So, a virus is a bit larger, actually. If you check a virus size, and while I'm talking to you, I'm actually like opening at the background. So most viruses are 20 nanometers. So the virus is even smaller, 8-fold smaller than my nanoparticles.
Really? They're smaller, the viruses?
Smaller than the particles, yes.
Wow.
And it depends on the kind of virus, right? You have retrovirus, which are a bit bigger. While I'm talking to you, I'm trying to there is an amazing Nature image that somebody just published that he compared nanoparticles to corona. Wait. Yeah, so I don't find it now, maybe I'll circulate it later. When you see the two, wait, you know what, I have it on my phone because I just texted it to one of the professors in Rice. We had a good discussion. Give me a second.
Sure.
I promise I'm not texting my wife. Wait, here you go. Let's see if you can see it. It will be easier. Okay. Wait. Okay. This is nanoparticle.
Okay.
The yellow one. This one is the virus. If you compare the nanoparticle versus the virus, you will understand that we are more than, I will say, 3,000 years behind nature. So nature is much better than us. He managed, the gray one is the virus. He managed to pack this genetic cargo in the most admiring engineering way. And again, I will text the image, and of course, Karen and Robert, please share it with everybody. It's not mine, it's Nature publication, and the author is, it's Moderna basically released it in their press release. But, you know, I basically learned from Moderna. So what I'm going to do is, you know, everybody asks me, Assaf, what will be your next lab? So my next lab probably going to be disease-oriented nanotechnology or disease-inspired nanotechnology. Because as I told you, nature is the best engineer. He developed the white blood cells, which I'm now engineering. He developed these viruses that are super effective. So all I'm going to teach my students is, hey, stop for a second, look what's happening in our body, try to use it for your tools during engineering.
Very interesting.
That's my thing.
Well, it has a good tradition, you know. Here, where I grew up in Dayton, Ohio, there were some guys who watched birds fly. That's nature, and from that they figured out how to fly, right? How to make an airplane.
I love it.
Assaf, at your teachers at the Technion, I mean, where did you get the desire to pursue cancer as your target, your attack of cancer? What motivated you? Was it before you got to the Technion, or was it at the Technion, or how did you get to where you are now?
So first of all, I would say thank you because nobody asked me this question. It just happened to be. But when I was 7 years old, my niece had leukemia, and she suffered a lot. She was the first patient in Israel that were transplanted by bone marrow. And since the age of 7, I knew that I will do something against cancer. So that's the inspiration for cancer. As I told you, Terry, at the beginning, spoiler, motivation.
Yes. Right.
The second thing I will add is that following your question, Robert, and I will give you a lot of credit to Texas as well, my eyes opened a bit. So saying we are fighting cancer, it's important. There is a huge hospital across the street, MD Anderson, that does that amazingly. But people dies more than cardiovascular diseases, heart attacks. People dies more than from influenza. But for some reason, we don't go to this direction. So science always try to say, we fight cancer, we fight cancer. Maybe we need to fight sepsis. Maybe we need to fight local inflammation. Maybe we need to fight liver fibrosis, right? And what I start to understand, again, very young, maybe my opinion will be changed next year. The main things that all of these diseases are sharing is inflammation. So if we will manage to hunt inflammation, basically we just need to tweak the car a bit. So that's what I'm going to do. I'm not going to do only this. I'm going to do also other inflammatory diseases. And you know, in my topic, there is also Rett syndrome, but let's keep something for the next talk, right?
I know, Susan, I know you have a question. Go ahead.
So assuming that you have the delivery system, how will you, I mean, what's the research and how do you go into the individual patient and how much of the anti-inflammatory they need so they don't get a storm? What's the process that goes on that you're going to use to bring it to the bedside and individualize it for those patients? Because every patient's going to be, different, and we don't need any more Geisingers, dying out there from something that's not going to work for everybody.
So in a second, I already can understand that you are Dr. Bowman, right? That's a physician question, so I appreciate it. Did I write? Am I right?
The developer. So,
okay.
Yeah,
Mossad skill in a second.
It's going to have what the dosage is.
So what I will say is like that. So we're testing two hypotheses. The first one, we're going to use the patient-owned blood. So basically, I need half a liter of the patient blood to make these nanoparticles. When we tested antibodies that were presented against our particles, we didn't see any IgGs nor IgMs. So basically, after 2 doses of injections, we did not see any immune reaction to our nanoparticles. And I will also add to it that our particles circulate for at least 72 hours. So after 2 injections, 10 days separate, of course, because we want to see the peak of the second antibodies, we did not see anti-inflammatory markers. And of course, we didn't see any anaphylactic shock due to, something for sure. So if we use the patient's blood, we didn't see something. But now comes the question, what if you cross-correlate, right?
Right.
And I don't know. That's something I'm working on it now. So we are doing it in large animal models right now, actually. And again, spoiler, I'm going to do it for cardiovascular disease. So basically, we are trying to sense inflamed endothelia that lead to atherosclerosis plaques in a very early stages. So this nanoparticle has contrast agents that I cannot elaborate on because it's part of patents that we are writing now. We inject them once a month, and basically, if we see accumulation of them next to the plaque, we know that plaque is forming. So we can basically prevent heart attack.
I see. Okay.
Let's see.
Thank you.
Thank you.
Yes. Are there any other questions out there? All right. Well, first off, we had a wonderful, intimate evening with Assaf. And Assaf, I can't say thank you enough for being with us tonight. It was absolutely sensational.
It's a pleasure, guys. Thank you for inviting me.
The people who aren't here, we'll have a recording for them, we hope, and we will have you back for this Dallas, Houston audience again in the future. Assaf has been one of our most distinguished graduates in a number of years, and he has been doing so many wonderful things. And we are hopeful that he's going to return to the Technion. And one of our biggest goals is to make sure we don't lose people like Assaf to places like Houston. So he's got about 400,000 Israelis and Technion supporters beating on his head to make sure he comes back to Israel for us. And I'd like to encourage you, you know all about Technion, everybody on this call, but the main thing is that recruiting people like Assaf is a real high priority for us. And of course, with his work that he's doing with COVID and the entire group of COVID labs at the campus, 50 of them in number. And they all operated even when the campus was shut down, ranging from mass development to drug delivery, to turnaround testing, breathalyzers, you name it, it's all there. And if you want to stretch your dollars further than they go in the US, support the Technion's COVID research or faculty recruitment area. It would be immensely helpful for us. And thank all of you for being here this evening. We appreciate it so very much. There'll be programs coming in the future.
Autonomous patient carts developed in the mechanical engineering department for the hospitals.
Thanks for putting that there. Forgot about that.
That's all.
Anyway, we will have more programs, and there'll be a couple of webinars coming up that'll be more national, but we're going to continue the local ones as well. And we will keep you posted on that. And we will update you in the near future when the next local one's going to be, as well as when the next national one will be too. So thank everyone. Thank you all.
Thank you.
Thank you. And just one more comment, and that is that I think that Assaf is a wonderful example of the future direction the Technion is going in terms of human health. Just like Assaf said, it's not just about cancer, it's about a lot of different diseases and human conditions that we can address with the kinds of interdisciplinary research that Assaf is an example of. So, you know, you know, Assaf is what we can expect more of in the future. And thank you, Assaf, for being such a wonderful spokesperson for the bright future that we will have.
You defined an important need that I hope the Technion works on, and that would be an airborne transport system between the laboratories and the hospital downtown. I assume that's what you're talking about. That would get you from one place to another in 5 minutes.
Oh, my motorcycle? Oh, I'm going to buy a drone, right?
Exactly, something, an autonomous transportation system that will carry people. Thank you, Assaf.
Everybody, have a wonderful night.
Have a good one, guys. Ciao.
Bye-bye.
Good to see everybody. Bye.
Bye.
Bye.